Thank you, everyone, and for joining us for what is now the last seminar in this particular Human Developmental Cell Atlas seminar series. We started the series on lung development in June 2020, and including this one, we've gone through 8 so far, all of which are or will be available on YouTube and Bilibili. So this is the pre-implantation, fertility, and gestation developmental seminar. Our guest speakers are Jean-Léon Mott and Wolf Reich, and there'll be 20 minutes each. And our panelists and a great discussion for the panel that's planned is with Esther Posfai and Frederick Lanner. And that'll be 45 minutes, including the audience questions. And we would love you to submit those questions. And we'll tell you how right here. So the slido.com HCA DEV 2021, if you're able to go there, or you can scan the QR code on your phone. The QR code should show on each slide in the upper right-hand corner in the next few slides. So you'll be able to scan that. And at the questions at the Q&A, if you go to the Q&A tab on Slido, you'll be able to select and you can ask your questions and also upvote questions as well. You can also add ideas and, to the right and also a poll. You can use the thumbs up on the questions and thumbs down, whether or not you believe a question is important or not. And the votes with questions with the most votes would be prioritized and hopefully being offered to the speakers and panelists. So for the ideas section I mentioned, you can share resources, any kind of ideas you might have under the ideas tab. And we would like to hear from you. There's a polls tab on the Slido, and you should be able to go to there and actually get some information on what you'd like to get out of the seminar. So if you join at slido.com HCA Dev 2021, you can start adding these in, and we should be able to start seeing these. So I'd like to welcome Jean-Léon Mott for his talk, and I'd like to introduce you to him. We're very fortunate to have him from the Institut Curie in Paris. His lab uses microscopy, genetics, and biophysics tools to understand the forces controlling early development of mammals, including humans. So I'll leave it to the speaker.
Thank you very much, Diane. I will share my screen, and I will ask you to tell me if you can see things properly. Do you see my slide?
Yes, looks good.
And you can hear me as well, and you can see my cursor too.
Yes.
Excellent. Okay, then I'm all set. Okay, so thank you very much. And I would like now to tell you about the mechanics of human embryo compaction. So we are interested in the early development of mammals, and the pre-implantation phase of development gives us a unique opportunity to study human development, because we can take a human embryo and put it in a dish and observe its development during the pre-implantation development, a live human embryos. And so here is a human embryo growing in a dish at the Hôpital Bichat. And you can see the divisions which continue on and on for the first 5 days or so of development, until it forms this structure here, which is called the blastocyst. The blastocyst is what's going to implant the embryo into the maternal uterus. And so we know how to now culture those embryos in vitro since the late 70s, and we got very good at this. And in a country such as France, actually, up to like 3 to 4% of birth are done through IVF technologies. So it's becoming more and more common, actually, for babies to be born this way. And even though we got much better at culturing those embryos, we are still not very good at knowing which embryo will make it after transfer. So typically, the kind of success rate that you get after transferring a human embryo into hopefully a mother, only a quarter of the embryo will succeed in implanting and will lead to live birth. So any additional knowledge that we can get about human pre-implantation development will not just help better understanding our own development, but also could help assisted reproductive technologies. So what you could see in this movie, and you can see it here on this snapshot, is how the human embryo in 5 days goes from the zygote stage until the blastocyst stage. And so it reaches the blastocyst stage by undergoing a series of cleavage divisions, which are divisions without cell growth, the cellular volume of the entire embryo remains fairly constant throughout pre-implantation development. It's only after implantation that there's actual growth of the cellular volume. There are a series of morphogenetic movement, which I will talk about in a second, and differentiation which start to occur starting from about the 8-16 cell stage. You can see in this blastocyst that there are two distinct cell types, the trophectoderm, which is in orange, a squamous epithelium, which forms an envelope around a fluid-filled lumen in green called the blastocell, and a cluster of pluripotent stem cells, the inner cell mass, which will give rise to all of the cells of the human body. The trophectoderm, its job is to implant the embryo and to contribute to extra embryonic tissues of the placenta, but not to the embryo proper. To shape this architecture, which is very important for the future development of the embryo, without this specific architecture, at least we definitely know from the mouse that if you affect morphogenesis, you will not get proper differentiation of the two lineages and you will not get implantation of the embryo. To get this morphogenesis of the blastocyst, there are different steps, which we like to cut them in 3 steps. The first one being compaction. Compaction basically just makes the embryo more compact, and we'll talk about it in a minute. And then there are there is the internalization of some of the cells of the embryo. So initially, all cells are in contact with the outside medium, but eventually, some of the cells are completely surrounded by neighboring cells. They do not touch the outside medium any longer. And this is very important for triggering the signaling cascade that will lead to the formation of the two lineages, the inner cell mass and the trophectoderm. And the last step, the last morphogenetic step, is the formation of a lumen, the blastocell, which will separate physically some of the trophectoderm cells and some of the cells of the inner cell mass. And this is important for creating a new interface, which will further help the differentiation of the inner cell mass into primitive endoderm and epiblast cells, and of the trophectoderm into mural and polar trophectoderm, which is important to define the site of implantation. So we are very interested in morphogenesis because it is important for the embryo to differentiate and to implant. Also because morphogenesis is the main criterion used by clinicians to assess the implantation and developmental potential of human embryos. They look at the embryo, they look at its shape, and this is more or less the only thing that they can look at to select the embryo. So we need to understand better what shapes the human blastocyst, because we need to understand better what shapes the human blastocyst, and if you want to understand what shapes an object, you have to start considering the forces which are in action, and we'll get into that in a minute. So let's focus for today on compaction, which is the very first morphogenetic movement that we do. So here is an embryo before compaction, and you can see that in about 10 to 20 hours, the embryo is becoming rounder, more compact. The surface of the embryo becomes smoother. The cell-cell contact are getting larger. You cannot distinguish single cells anymore within this embryo. And this is essentially a developmentally regulated adhesion process. This process can fail, and this has been studied. The way the clinicians are doing is that they grade the embryo by eye, and they say that this one here is not compacted, this one is poorly compacted, and this one is well compacted. And when they do so, they find that the embryos which are the most compacted have better chances at implanting and giving rise to a live birth. It is not just the magnitude of compaction, but also the timing of compaction, which is important. Embryos which compact before 80 hours after fertilization have better chances of implantation. So the magnitude, the timing of compaction, both of them are important to maximize the chances of compaction, of implantation, sorry. Sometimes what we find as well is that some embryos will be fully compacted with all of the cells equally well compacted. And sometimes some of the cells are excluded from the compacted mass of the embryo. You can see here, for example, this cell here on the left and those two cells on the top. And so these are called partially compacted mola. I like to call them unequally compacted embryos. Sometimes those excluded cells will contribute to the embryos, mostly tissues of the placenta. And sometimes it will be entirely excluded from the blastocyst and will not contribute, not even to the placenta. What's interesting is that those cells tend to be aneuploid. They have ploidy defects and they are excluded from the embryo. And sometimes they go into the placenta, and we can find them in the placenta actually. So what's been proposed is that compaction is actually a mechanism by which the embryo is eliminating sick cells, cells which are not fit, which are aneuploid, and it's pushing them either completely excluding them from the embryo or putting them into the extra-embryonic tissues of the placenta. And we do not know what drives this exclusion. So what I would like to tell you today is about what drives human embryo compaction. And so, as I mentioned, if you want to understand what shapes an object, you have to take into account the forces which act onto this object. And in animal development, those forces are mostly of mechanical nature. So if you consider the forces which could be compacting an embryo, and so if we consider just two cells, we can assign them two types of interfaces. We have the cell-cell interface and the cell-medium interface, or the non-contact interface. And those two types of interfaces will have different surface tensions. There's a surface tension here in red, gamma cm, the tension at the cell-medium interface, and it tends to make this interface smaller. And there's a tension at the cell-cell contact in green, gamma cc, which tends to make the contact smaller. The balance between those two tensions will define the angle of contact defined by this contact. And so now if you want to compact the embryo, so if you want to make the angle of contact larger, you can either pull harder on the edge of the contact, increase gamma cm, or you can relax the cell-cell contact by decreasing gamma cc. And so both of them are possible, and different cellular mechanisms will be involved in those different phenomena. For example, what is mostly proposed in the literature is that compaction is mediated by an increase in adhesion, an increase in the activity of the adhesion molecule, and that will lead to the decrease of this tension gamma cc. So to understand how the embryo compacts, we need to measure the surface tension, and the way we do this is by using micropipette aspiration. So you have here a micropipette, which is schematized on this diagram, and we apply a pressure at the surface of the cell until we can deform it with a specific deformation, which is a hemisphere. And at this point, we can relate the tension gamma cm of this cell to the pressure that we have set into the micropipette and the shape of the pipette and the shape of the cell by using this Young-Laplace equation. Once we have measured the tension gamma cm of this cell, this cell, this cell, and this cell, we can apply another force balance equation, which has the exact same assumptions that the cells are behaving like a liquid-like droplet. We can apply the other force balance equation, which I mentioned, which relates the tension gamma cc at the cell-cell contact, which we cannot access by the micropipette, and the tension of the two contacting cells, as well as the contact angle. And so using those two equations and a micropipette to measure multiple cells and a microscope to measure the contact angle, we can get all of the tensions of all of the interfaces of the embryo. And because this technique is non-invasive, as you will see, we can actually measure the tensions over time. So this is a mouse embryo. We had published this in 2015. The embryo is being measured with this micropipette as it is compacting over the time course of about 10 hours in the mouse. And you can see the embryo is nicely compacting, and we have measured it about 40 times. So now we have this approach, and all we need are human embryos. And so we could get recently the permit to get those human embryos, which are donated to research in France. And we set to map the tension during compaction. So this is a human embryo grown in the lab. We have stripped it away from its zona pellucida, the eggshell, which you will not see very much in this presentation. We removed the eggshell around the embryo, and we wanted to make sure that the embryos can grow in the lab under the microscope with a pipette aspiration and without zona pellucida, which could be a challenge. And you can see this embryo developing, dividing. The pipette, you could see its shadow popping up at some point. The embryo is nicely now compacting and starting to form the blastocyst. So we make sure that the embryos that we measure develop until the blastocyst stage. So now what happens to the contact angle and to the tensions? So here is a human embryo. It's compacting. So here it's about 12 hours later. And you can see the contact angle on the left. The angle of contact is growing from about 60 degrees to 150 degrees plus. These angles are similar to what we observe in embryos which are grown at the clinic without any manipulation, other than the one that they do at the clinic. And they are similar also to the contact angles that are observed in mouse embryos. So there's a similar compaction in those embryos which are in the lab, the one of the clinic and the one of the mouse. At the same time, as the contact angle is increasing during compaction, the surface tension gamma cm at the surface of the embryo is increasing from about 500 piconewton per micrometer to above 2000 piconewton per micrometer. So there's a fourfold increase in tension at the cell medium interface. At the same time, the tension along cell-cell contact is not relaxing, which would favor compaction. It's staying fairly stable. So what seems to be driving compaction would be this concomitant increase in tension gamma cm and the angle of contact. And so you can then relate those two parameters and check the correlation. So there's a good correlation between the surface tension gamma cm at the cell medium interface and the angle of contact. So it can explain compaction. The tension gamma cc at the cell-cell contact is poorly correlated with the angle of contact, and it cannot explain compaction. So what explains compactions is this fourfold increase in tension at the cell medium interface. Now, what drives this tension increase? So what's found in many animals, including the mouse, is that what controls surface tension at the cell medium interface is the contractility of the actomyosin cytoskeleton. So what we do is we can inhibit the contractility of the actomyosin cytoskeleton using drugs. The drug we have used here is called ML7. It targets the myosin light chain kinase, which is required to activate non-muscle myosin 2 and actomyosin contractility. We want to make sure that we are not damaging the embryos too much. So what we did is we first incubate the embryos in medium which contains the carrier of the drug, ethanol, for 45 minutes. We want to make sure that we do not affect cell division, compaction, and so on, and we measure tension. Then we transfer the embryos into the drug, and after 15 minutes, we start measuring the tensions of the embryo for no longer than 45 minutes. Then we wash off the drug, and we want to watch development until blastocyst stage. And so this is what happens when you do this experiment. The embryos start with a high tension and a high contact angle. Then you put them into the drug, and the tension drops down by about threefold, and the contact angle is also reduced. So if you look into a bit more details, what you see is that, so as I said, the contact angle is decreasing by about 30 to 40 degrees. The tension gamma cm at the cell medium phase is decreasing by threefold. The tension at the cell-cell contact is also decreasing. So there seems to be a significant amount of contractility at cell-cell contact, which opposes compaction. And in this case here, by decreasing the tension gamma cc, we actually favor compaction. But because the decrease in gamma cm is larger, we have a negative effect on compaction in the end. What we conclude from this is that actomyosin contractility is required for generating tensions in the human embryos and for driving compaction. So this fourfold increase in tension is mediated by actomyosin contractility. So we can now compare with different species. We had done this experiment in the mouse, and in the mouse, there's an increase in gamma cm and there's a decrease in gamma cc. While in the human embryos, there is no decrease in gamma cc and there's an increase in gamma cm. So qualitatively, compaction in human and mouse goes in the same way. There's an increase in tension specifically at the time of compaction, but quantitatively, there's a slightly different strategies between the mouse and the human. The mouse is relaxing its contact a little bit and increasing its tension gamma cm by about twofold. The human embryos entirely relies on the increase in tension at the cell medium interface. We started looking also at other species, so rabbit and cow. And rabbit and cow embryos, we have very few measurements. These are just very preliminary measurements. It looks like the cow embryo is also seeing an increase in tension during the compaction process, and we need more measurements in the cow and in the rabbit. What I would like to point your attention at is that we can actually compare those different species because we obtain measurements which are in a physical unit. They are absolute values in a physical unit, so we can directly compare those species. And what you can see is that the cow, the mouse, and the rabbit, they have tensions which are in the range of hundreds of piconewton per micrometer, while the human embryo has tensions which are much higher in the range of thousands of piconewton per micrometer. So there seems to be a lot of mechanical stress at the surface of human embryos and not in those other species. We do not know what this means. So now that we understand a bit better what drives human embryo compaction, we can try to see what prevents it? And so we looked into embryos which fail compaction, and because there's no proper definition, no quantitative definition of failed compaction and so on, we had to define it ourselves. And so we consider an embryo which is normally compacted when all contact angles are higher than 140 degrees. And we consider that compaction has failed when cells are still dividing, but no contact angle reaches 140 degrees. Now, those embryos could fail compaction either because they fail to increase gamma CM or because they increase gamma CC abnormally. What we find is that embryos which fail compaction, they do not increase gamma CM. So there seems to be a defect in the ability of the embryo to increase gamma CM, presumably because of a failure to increase actomyosin contractility. What we also see, which is less dramatic, are those cells which are excluded from the embryo with this unequally compacted embryo. And we define this by having an embryo which is nicely compacted with angles higher than 140 degrees. But cells which are excluded show a contact angle lower than 120 degrees. Now, why do these cells get excluded? What we find is that before compaction, there's no obvious difference between the cells which will be excluded and the cells which will not be excluded. We have a nicely compacted mass here in the center, and some cells which become excluded. Obviously, the contact angle does not increase during compaction. That's what defines them. But what we see is that the cells which fail to increase contact angle, they initially already have a lower surface tension than the cells that will eventually increase their contact angle. So they already have a defect in tension before they get excluded. And then as the embryo is compacting, the cells which compact increase tension, but the cells which do not compact do not increase tension. So the cells are excluded because they fail to increase the surface tension, presumably because they fail to increase contractility. What we also see is that the cells which are not excluded, they tend not to change gamma CC too much. In some cases, it can decrease a little bit. This is not significant in terms of statistics. We need more data. But the cells which are excluded, they also tend to increase tension at their cell-cell contact. And I mentioned that there's quite some residual contractility at the cell-cell contact of human embryos. And so there could be actually an ectopic contraction of the cell-cell contact. So the cells could be actively excluded. And so this is what we are looking into at the moment. We are looking into the localization of the actomyosin cytoskeleton. And you can see the compacted part of the embryo in the center, which has an actomyosin shell around it with not so much myosin at the cell-cell contact, while there is some actomyosin at the interface between the excluded cell and the compacted part of the embryo. So what we think is that there could be some kind of active exclusion of the cells, which potentially could be aneuploid. With this, I'm actually concluding. So we wanted to understand what drives human embryo compaction. And what we found is that there's a fourfold increase in surface tension at the time of compaction. This is what drives compaction, and this requires actomyosin contractility. Failure to increase tension explains why some cells and some embryos fail to compact, and this could explain how some cells are excluded from the embryo because of their aneuploidy, for example. This is a project which is carried out by a medical doctor in the team, Julie Firmin, and this is a collaboration with Catherine Patra, who is a medical doctor who is in charge of the fertility clinic at the Hôpital Cochin. The embryos from cow and rabbit were obtained from Véronique Duranton, and we are collaborating with the lab of Hervé Turlier, who is a physicist who is developing theoretical models of morphogenesis, and we hope to be modeling human embryo development at some point. Thank you.
Thank you, Jean-Léon. And we'll keep our questions to the panel part of the discussion. It is now my great pleasure to introduce Dr. Wolf Reich. He's the director of the Babraham Institute in Cambridge, UK, and the head of the epigenetics program. He's an honorary professor of epigenetics at the University of Cambridge. He's also associate faculty at the Sanger Institute, where he's a founding member of the Center for Single Cell Genomics there, and is also affiliate faculty at the Stem Cell Institute. He's a member of EMBO, a fellow of the Academy of Medical Sciences, a fellow of the Royal Society, and a member of the Academia Europaea. His lab focuses on epigenetics, especially in epigenetic reprogramming during mammalian development and its role in stem cell biology, inheritance, and aging. So thank you, Dr. Reich.
Thank you very much, Leon. Can you all see the screen and everything? Everything?
Yep.
Everything good? Excellent. Thanks very much, Diane, for the kind introduction, and thank you for having me. It's a great pleasure to join this fabulous seminar series. I only wish I could do it in person, like probably every one of you wishes, and maybe in the future we can do that again. So just to introduce ourselves a little bit, we're located at the Babram Institute in Cambridge, and we are part of a really lovely grouping of excited epigeneticists, people that like to understand epigenetic principles that underlie development, stem cells, aging more recently as well. So it's a wonderful environment. Please come and visit us when you have a chance to do so. So in the lab, we're particularly interested in three things, which is global epigenetic reprogramming at the beginning of life, so to speak, pretty much immediately after fertilization and also in germ cells, and increasingly the link with reprogramming and zygotic genome activation, which is this amazing phenomenon whereby the embryonic genome all of a sudden springs into action transcriptionally quite early on in mouse and a little bit later in human embryos. Then we're very interested in cell fate diversification during gastrulation and early organogenesis, and that's where we're applying a lot of our single cell technologies, which I'm going to tell you about a little bit today. And then more recently, we're interested in the other end of development, so to speak, which is aging, and how does the epigenome degrade during the aging process, and what are the prospects for rejuvenation of the epigenome, if there is such a thing. So today I'm going to focus entirely on cell fate diversification during gastrulation. And just before I get into the main sort of topic, which is epigenetics, I just want to give you a couple of snapshots on our recent technology development. And one of which we're getting into now is the spatial mapping of single cell information. So previously with our consortium, established atlases of mouse development, single cell expression atlases of mouse development. But of course, you dissociate the cells, you lose all the information about the spatial context that this transcription occurs in. And of course, that is important because eventually we'd like to understand the signaling principles, cell-cell interactions, the kind of stuff that Jean-Léon just talked about, and how does that affect cell fate in the three-dimensional space of the embryos. So with our consortium and the wonderful collaboration with Long Kai at Caltech, and using his Soq-Fish technology, we made a spatial single-cell atlas of mouse development, which uses 353 genes, a sequential in situ hybridization, so to speak, called Soq-Fish. And this results in beautiful expression patterns of individual genes, but also by then importing all of the information that we have, 10,000 genes or so in the transcriptional atlas into the spatial atlas. We can identify cell types, we can identify lineages. You can go to the website, there's a Shiny Web app, you can look for your favorite gene, etc., etc. So please use the resource and give us feedback of how it works for you. Then in a further evolution, what we're trying to do as a next step is to map epigenetic, single cell epigenetic information onto the spatial expression atlas. And just show you one example here, which I'm really, really excited about, which basically shows in a multi-omics single cell format, the expression here in the brain, in the forebrain and midbrain, of a particular transcription factor. And then next to it, the chromatin accessibility to the motif of that very transcription factor that is expressed in those tissues. And that, to me, is super beautiful and sort of tells us a little bit that maybe in time we will be able to import much of the multi-omics epigenetic information that we now have into the spatial atlas. So super excited about that. But underlying, obviously, we are epigeneticists. We're interested in what are the transcription factors and the epigenetic programs that determine cell fate during gastrulation, which we at this point use as our favorite model system, so to speak. And this is our most advanced technology, where we can sequence 3 dimensions within each single cell, which is the transcriptome, the methylome, and chromatin accessibility. And what we've done here is to try and understand, using this triple-omics, single-cell triple-omics method, to understand what are the underlying epigenetic principles of germ layer allocation. So the allocation of the mesoderm, the ectoderm, and the endoderm in the mouse embryo, which takes place from day 6.5 to day 7.5 in development. And the answer, the global answer, is very, very simple. There's nothing really interesting going on in promoter epigenetics. There's nothing going on in transposons. In other regions, regulatory regions in the genome, the main action is in enhancer epigenetics. And so basically what you find is that thousands of enhancers, ectoderm-specific enhancers, become demethylated or are demethylated in the ectoderm, but not in the endoderm. And thousands of these enhancers, in ectoderm enhancers, are chromatin accessible in the ectoderm, where those genes are obviously transcribed. So the pattern globally is very, very simple. Enhancer epigenetics defines the 3 primary germ layers in the mouse embryo. But what is more interesting is to ask how, in time, the lineages get to that point. So because we now have a time-resolved series where we started, in fact, the epiblast at 3.5 days of gestation, and then all the way to 7.5 of days of gestation. So I'm just going to briefly show you again the global pattern here, just in a different way of showing it, the global pattern for the enhancer epigenetic landscape. So basically in the ectoderm, after gastrulation, all the ectoderm enhancers, thousands of them, are demethylated, that's the red curve, and are chromatin accessible. And of course, in the ectoderm, the endoderm enhancers are not demethylated, are not chromatin accessible, and the same goes for the mesoderm enhancers. I've just rephrased and show you in a different visualization the same global pattern that I told you about before. But how does these patterns arise in time? And this is where we got a huge surprise, actually, because if we look very early, then the endoderm enhancers, they are blissfully asleep. They're not thinking about yet about making gut at this early time point. The mesoderm enhancers are asleep, epigenetically speaking. They are not thinking about making muscle or heart at this early time points. However, the ectoderm enhancers are quite different. They're already a little bit demethylated. They are already chromatin accessible. And this is 3 or 4 days before these genes begin to be expressed to make brain and skin in the future. So these enhancers, ectoderm enhancers, but not mesoderm and endoderm enhancers, are said to be primed in this very early epiblast structure, undifferentiated epiblast, which is quite surprising. So then you can see this a little bit better here now, 1 day later, because now you'd have this, the genome experiences massive de novo methylation. So the red line, methylation line, has moved up. Here you see the demethylation, hypomethylation, still of the ectoderm enhancers. The genes are not expressed yet, but the enhancers are thinking about future activation of these genes. So what's going to happen with the endoderm and mesoderm enhancers? Eventually, they have to wake up and make gut or muscle, of course. And they do so very, very precipitously as the cells ingress into the so-called primitive streak, are exposed to different signaling environment in that context. And all of a sudden, thousands of mesoderm enhancers lose their methylation, become demethylated, become chromatin accessible. And this, in part at least, from a mechanistic standpoint, is driven by the TET enzymes that demethylate DNA. We know that from mechanistic studies. So what we have found, we think, in the mouse, and I'm going to show you a snapshot of human development in a moment, is that the three lineages, whilst they're driven by enhancer epigenetics, they do so in a very hierarchical or asymmetrical model, whereby the ectoderm enhancers are awake at a very early stage and in the undifferentiated epiblast, already being primed or thinking about needing to be active in the brain or in the skin, whilst the mesoderm and ectoderm and mesoderm and endoderm enhancers are blissfully asleep at that point in time. But then the cells take a sharp right turn into the primitive streak, are exposed to different signaling environment, and all of a sudden remodel thousands of the enhancers epigenetically to activate these genes in a very acute kind of way. So the questions that we're now thinking about in this context is that we would like to increase time resolution and throughput. These methods are still quite time consuming and low throughput. So that's going to be important, and we're working on that. We're asking the question of how general is this hierarchical cell fate model? Is this something that's limited to gastrulation, or is this also important for adult stem cell differentiation, for example? Do they have these hierarchical ways of doing things? Very interested in that question. We're applying our insights into human development, collaborating with Jenny Nichols and also Frederick, who's on the panel. And I'll show you a snapshot of that work in a second. And we're very keen to identify DNA binding proteins that are involved in this very early lineage priming that is so important maybe for some lineages to be established. And then more mechanistically speaking, we're interested in the mechanisms of demethylation and also the role of de novo methylation, which is still very mysterious in development. So just a couple of quick snapshots before I close. So is there such enhancer priming also in human development? We're very, very excited to find out about this. And that there is the potential here for good news. If you look at this human ES cells here and human neural precursor cells as an initial neural differentiation product, so to speak, then you can define in the neural precursor cells the active enhancers. Here they are. And then you go back to the human ES cells and you find that a quite large proportion of those active enhancers are actually already primed in the cells in which these genes are not yet expressed. And also another large proportion, roughly equal size actually, is not primed at this early stage. So what distinguishes primed from non-primed enhancers and what developmental programs they control in human development is of great interest to us. And here you can see the DNA methylation. This is a different mark here, which is K4 monomethylation, which is a mark of enhancer priming. Here you can see again that the primed enhancers are hypomethylated in relation to the non-primed ones. And even in pre-implantation embryos, very excitingly, the epiblast, the early epiblast, ICM not so much, but the early epiblast already shows signs of demethylation of these so-called primed neural enhancers in human development. So very excited about this. And then the final snippet, I think, yes, the final snippet I want to show you, this is more of our mechanistic work, is trying to identify basically proteins or transcription factors that prime regulatory elements in early development. We have done this so far in mouse. We're also working on human factors right now. And what we found is that two proteins, DPPA2 and DPPA4, are very important in directly targeting polycomb and trithorax proteins to so-called a subset of so-called bivalent genes, gene promoters in development that are marked both by activating K4 trimethylation and repressing K27 trimethylation marks. If you don't have these proteins in a knockout, basically, then you lose bivalency, you gain DNA methylation at these promoters, and then during differentiation, and this could be many days in the future in development, these developmental genes cannot be properly activated, and the cells cannot properly differentiate, and the embryos cannot properly differentiate at quite late stages sometimes in development, which is fascinating, I think. So here I'd like to thank all of the wonderful people in the lab. This seems ages ago. This was our lab retreat in 2019, which I only dimly remember myself, but it was a real lab retreat. Wonderful collaborators we have, in particular in our so-called gastrulation consortium, and wonderful funders that allow us to have fun in the lab. And thank you very much for your attention, and happy to discuss.
Thank you, Wolf. That was really, the last two speakers are really wonderful, and that retreat looks great. Yeah. So we're running a little early, but that's great because that'll give us more time to get to questions and also introduce our panel. So I would like to introduce our panelists. Esther Postfai, she joins us from Princeton University in the United States, and her lab studies early embryo formation with a focus on understanding molecular and cellular dynamics using live imaging. And from our conversations, she's a big proponent of using multi-omics to try to understand embryo development, as probably of many people on this in this conversation. And Frederick Lanner is a panelist, and Wolf introduced some of his work with him earlier. He hails from the Karolinska Institute in Sweden, and his lab actually established a transcriptional atlas of human pre-implantation development, which is also one of the conversations that Wolf talked about. And also he continues to work in post-implantation development now in pluripotent states, X chromosome inactivation, and how the human embryo prepares for gastrulation. And we were interested in hearing from the panelists, maybe a 1 or 2 minute conversation on what they think the important topics are in this field. I don't know, Esther, do you want to go first on this?
Okay, sure. We're diving in.
Yeah.
Okay. Hi, everybody. I think what we wanted to start out with is a little bit talking about the importance of integrating multiple approaches for the study of early embryo development. And I think Wolf has made a pretty good case for multi-omic techniques. And I guess I just wanted to put in a few words for studying embryo development using live imaging, which is along the lines of Jean-Léon's presentation, and which is also the main approach that my lab is using. So experimentation with pre-implantation embryos goes back over 50 years. And since then, a lot of molecular profiling has been done. Mostly recently, maybe in the past 10 years, single-cell transcriptomics has especially given us really a rich and detailed insight into some of the players that are present during embryo development. And then also decades of functional studies generating transgenic animals and then knockouts and so on have told us which players are important for embryonic development. So this rich information that comes from these techniques is obviously a huge advantage, but there's also limitations, and that is that we are looking at dead things, meaning that we are looking at fixed snapshots, while of course living and developing cells and embryos are very much dynamic. So essentially the problem is that if you're attempting to reconstruct time courses from snapshots of different cells at different times, you cannot distinguish between temporal changes from cell to cell variability. And this is especially important, I think, to consider in the case of developing embryos, mammalian embryos, where there's a lot of cell-to-cell variability, and the dynamic crosstalk between these variable cells is what's going to determine what a cell does and what a cell becomes. So molecular profiling and knockout studies are fantastic to generate a parts list, but the challenge I still think still remains that we need to figure out how exactly these parts and pieces play together in space and time. And I guess that's my pitch for where live imaging comes in to really fill this gap. And then, of course, another huge advantage of live imaging is that it can be quantitative if done properly. And then I'll hand this over to Jean-Léon here, who I think is going to say a few words about the importance of making quantitative measurements for studying embryo development.
Sure. So basically what we try to do in the lab is we try to have quantitative methods, and obviously quantitative measurements have broken through most of biology, hopefully by now. But I would like us not to stop there. I think it's important that biologists do not only just measure things quantitatively, but also try to get quantitative measurements in absolute values, because an absolute value, ideally with a physical unit, gives you much more information. Think about, if you want to compare the size of two objects, let's say the mouse and the human embryos. How do we know that the human embryo is larger than a mouse embryo? It's not because someone put them side by side. It's because someone looked at it with a microscope, which had a scale with a scale bar in micron, and could compare the mouse and the human embryos. And so having a physical unit can help ask different questions. And so this is also what we try to do. And I briefly mentioned the fact that the human embryos have this super high tension compared to mouse, cow, and rabbit. And this is only because you can get a quantitative measurement in absolute values. And I would very much encourage everyone to try to not stop at being semi-quantitative. Semi-quantitative is already very good in many instances. And of course, if you start to look at epigenetic marks, I don't know how much quantitative measurement in absolute value you can do. I don't know how many methylation you can count. But ideally, I think it would be good to also make an effort in this direction. Let's say if you want to look at the level of gene expression of whatever genes you're interested in, you can go semi-quantitative, but you can also do single molecule on a fish and actually count on a molecule if you wish, right? And there are some other techniques also to try to be more quantitative, but in absolute values.
Frederick, do you want to give your opinion?
Yeah, sure. I mean, these are all very exciting opinions, of course. I'm not of a different opinion, but I think my lab has been focused on trying to understand pre-implantation development. First in the mouse as a postdoc, and now the human in my own lab. Although we have a lot, of course, to learn about human pre-implantation development, we at least know when the first 3 cell types emerge. We know which genes are expressed, how they turn on. But when we go beyond the implantation, I would say we almost know nothing about how these cell types sort of mature, and also how new cell types emerge. New cell types that we don't really have emerging in the mouse. So we don't have an animal reference, at least in the mouse. So we don't know how the amnion is formed, at what point this happens. In the human, we have these peculiar extraembryonic mesenchymal cells starting to fill the blastocyst cavity just a few days after implantation. We don't really know even the origin of those cells. And also transcriptional profiles or markers of those cells are completely absent, or almost completely absent. And these cells are, of course, extremely important when moving into gastrulation and when preparing the embryo to gastrulate, which Wolf really beautifully sort of described in the mouse. But as we all know, gastrulation in the mouse and the human are 2 complete different scenarios, or at least it appears to be very different, sort of morphologically, and probably also on a regulatory level. So I think that's the big challenge, at least for that I see, trying to move beyond pre-implantation development and trying to build a better understanding for how the embryo implants, matures, and prepares for gastrulation. And I think extended in vitro cultures of human embryos is one way of moving there. There's more and more exciting developments, blasting developments when it comes to stem cell based models of early human embryo development that can become very powerful. We also need to be very careful about these models, because I think they can take us in the wrong direction also if we're not very careful. Yeah, I think I'll stop there for now.
And we can go to questions too, and also there's a question that was tagged for Wolf, but it really probably goes across most of the panel. From Martha Shabazi, she asked, the hypothesis of aneuploid cell elimination during compaction is very interesting. Since aneuploid cells are clearly found at the blastocyst stage, how do people think that this mechanism could work? Maybe just detecting complex aneuploids or specific aneuploids that are detected. How possibly do embryos eject these aneuploid cells? What kind of mechanism, I guess, would be used? If anyone has an insight into that or hypothesis.
I don't. So I'll leave it for someone else to pick it.
I can take it if you want. Yeah. Okay. So this idea that the cells are attenuated during compaction is something which has been proposed after basically checking the DNA of excluded cells and non-excluded cells, and some of those cells are excluded entirely from the embryo, so they are, I would say they're presumably dead, and some cells actually contribute to the trophectoderm. And so, what we find is that there are differences in tension between the cells that are excluded. And we have another work in the mouse which says that cells which are part of the trophectoderm are part of the trophectoderm because their tension is lower than the cells that will belong to the ICM. So I think this could be a good explanation for the exclusion of sick cells, if that was the question, Martin.
Can I actually ask something related? I think the question was going towards, can you speculate on the actual mechanistic link between a certain aneuploidity and lower actinizing contractility?
Yeah, so this is something, so if you look at, so you could imagine that, so Marta actually has some work on how losing specific chromosome can result in changing of expression of ectoderm, for example, so in the case of contractility and surface tension, actually, so what we find is that the tension is low already before the exclusion. And if you look at the literature on actomizing contractility, defects in actomizing contractility are often linked with defects in chromosome segregation. So actually, this lower tension could be the explanation for the aneuploidy itself. And so we are not seeing just the mechanism by which the cells are excluded, but also the cause of this aneuploidy in the first place. We have not formally identified that those cells with low tension have aneuploidies.
Because it's not that likely that the transcriptional profile would mark them out, or to be able, to be the basis of the sorting phenomenon that.
So the sorting in space?
Well, just, to eliminate the cells, basically. That's the question, isn't it? It's Marta's question.
Obviously, those differences in mechanics which sort the cells, they arise from differences in the components of those cells, right? Which you could spot with transcriptomics if they arise from a transcriptome difference. But if they arise from protein differences, then you will not see it.
So we have a couple more questions here. One of them is a general panel question from Bruce Arano on early lineage differentiation, the role of demethylation. How modifiable are these TET and maybe DNMT pathways by environmental agents, nutritional status, and maybe even high doses of pregnancy vitamins like folates? So that comes back to folic acid supplementation. So, which is obviously, or we think is related to healthy embryo development. So how modifiable are these pathways, Wolf, maybe by environmental agents, do you think? Like how much of an impact do you think you maybe could test or otherwise test in your models?
I think that's all. It's a very good question. I'm not an expert on this, I should say. Vitamin C is an important cofactor for the TETs, as is vitamin A, actually. And then there's some other cofactors that are known. And yes, you would expect that if you modulated the availability of those factors during culture periods, you could affect the epigenome, but it's a hard question because those effects are so global, and then we're talking about some very specific things. So there's a big black box in the middle here somewhere.
Is anyone else? So Gary Bader has also asked a general panel question of everyone, which is actually probably an entire, it encompasses this entire seminar. Is what are some of the major questions in the pre-implantation field, and how can current and future single-cell genomic technologies help directly address those? So it's in specific the single-cell genomic technologies. We did talk a little bit before the seminar about multiomics. That could be one. Or it could be, are there other types of applications that you could think of that might help address any major questions you have in your mind? Next steps or something like that.
I could say something about this. We talked a little bit before about this black box. So what's the end of pre-implantation development, and what is a measure for a developmentally competent embryo? And we don't know in human, and we even don't know in mouse, actually. We don't have the parameters to say that this embryo for sure is a developmentally competent embryo. I can give you a rough cell number for, like, let's say how many epiblast, primitive endoderm, and trophectoderm cells an embryo should have, but then if you deviate from these average numbers, where's the limit, for example? We don't know. And I think a lot of things that developing non-invasive techniques to study embryonic development, for example, by live imaging, and which in mouse we can combine with fluorescent reporters to then actually look at directly different cell lineages and cell numbers and organization. Combining these non-invasive techniques, and then also improving development of extended culture of embryos in vitro, and along the lines of work that Martha's doing, or Ivan Betsov in mouse embryos, to really have, if you have a reliable assay for functionally testing if these embryos can proceed and develop further, I think connecting the endpoint of pre-implantation to then post-implantation studies will be very important. And this is, I think, a current need in the field.
Yeah, I agree that sort of trying to link any type of sort of characteristic of the pre-implantation embryo, where we have a clinical opportunity of sort of analyzing that prior to transfer and see if the success rate of pregnancy or implantation, if that's at all correlated with the in vitro success rate of implanting and developing in our current models. I mean, if there is a correlation there, we have a completely new toolbox, I would say, to sort of try and fish out what is a good quality embryo, either by morphology or from a trophectoderm biopsy, or going for spent media or blastocell fluid and all these sort of techniques that have been adopted so far. Perhaps not with so amazing success yet, but if we can sort of link it to implantation and post-implantation competency in vitro, that would at least open up for new possibilities. And that could be, of course, also at the transcription level or genomic level, looking at mosaicism and so on. So I think that's an exciting development. But first we need to know that actually these in vitro models that we have say anything about the general competency. It could also be that our in vitro models are just quite poor still. So I think that needs to be addressed and looked at in some way.
From our perspective, if I could just add that, maybe I think what's particularly interesting is to ask how these mechanical forces and signaling changes then impact on the transcriptome and the epigenome. And there are some things that are sort of a little bit known, like the hippo pathway maybe is the the poster child to some extent. I don't know whether that's true in human, for example. Maybe you guys can comment on that, what's known about this. But I think that's really, you know, how the different single cell dimensions interact, basically. That's really important to understand better in the future.
Linking the structural aspect of cell-cell communication, this epigenetic expression. Someone who remained anonymous asked, I would like to know if cell-to-cell communication among the different structural cells in the embryo regulates epigenetic expression. And this would come into the whole idea of the cell-to-cell communication, but also the cell. I would think it's also the cell-to-cell stress that and the tension that Jean-Léon was talking about earlier. The link between that and epigenetic expression. If there's a link between those two.
So most likely, cell communication is controlling gene expression. That's a given.
And differentiation, you think?
Say it again.
Like in differentiation, like actually epigenetic changes and differentiation and things like that. Would that have a direct impact on the cell-to-cell tension and the communication between the cells? Environment.
What we know from the mouse is that the shaping of the embryo is very, and the positioning of cells is very important for the differentiation. So I don't know how much has been done on the specific epigenetic modifications, but I assume that since the cell types are becoming transcriptomically different, they have some epigenetic differences. And this results from cell-cell communication based on either you have a lot of cell-cell contact or a lot of cell-medium interface. That's in the mouse.
Mm-hmm. Any other comments?
So is there anything known about the hippo pathway in the human embryo?
I believe Kathy Nikon has a paper on that, showing that YAP localization differences are conserved and similar between mouse and human. So yes. And then Magda Zernica-Gutz has some papers on the polarization of the human embryo, which is kind of the upstream thing that matters for hippo or YAP localization. So it seems that at least the major players in the earliest cell fate decision between mouse and not are conserved.
Has there been significant work done in looking at embryos that may have chromosomal abnormalities consistent with life and compatible with life? For example, looking at everything we've talked about today in trisomy 21, 13, or 18. I mean, what could we learn from these embryos? Because they are compatible with life and people are born with these abnormalities. Is there anything that's unique? Because when we studied this ourselves for looking at, we did a study of development in embryos with very specific chromosomal abnormalities. We noticed that Down syndrome, I believe that the embryos that children would be born with Down syndrome, their embryos had much slower development. Although they were compatible with life, they developed a lot slower. Is there anything we can learn from those, do you think? Those kinds of studies? Or not?
Is there any chance we can unmute Martha Shabadzi, who was on that? Because that's actually her entire research area that look at different chromosomal abnormalities in human embryos and trying to culture them through implantation stages and looking at, I guess, what they turn into. But I can't say much more details than.
Yeah, so I mean, I can try to give an answer to that. So I think those embryos are actually very valuable. So like Esther said, we've been looking at aneuploid embryos with specific abnormalities. And first of all, our study really showed that it's very important to focus on specific aneuploidies and not just talk generally about aneuploidy, because of course, depending on which chromosome is affected, the outcome is one or another. And in this study, we looked at the development of trisomy 21, trisomy 15, 16, and monosomy 21 embryos up to day 9 of development. We just did a global morphological characterization of these embryos. And just looking at this global morphology, we were not able to observe any major abnormalities in trisomy 21 embryos. But of course, this does not necessarily mean that there could be other potential alterations there. And then also linking my response to something that Frederic mentioned before. So one of the objectives with this study was really to try to understand whether we could use these extended culture systems to look at developmental competency. And I think it is quite promising the fact that, for example, when we were analyzing embryos that reached the blastocyst stage only on day 6, they were developing further very poorly in vitro. And we know from clinical data that when embryos that reach blastocyst stage on day 6 are transferred, the success rate is lower. So we could actually recapitulate some of those findings in patients in our in vitro systems.
Yeah, I fully agree. I didn't mean to sort of criticize and say that there was no correlation. It's just that I think we need to sort of look at that further. And I also agree with your point that we can't look at aneuploidy in general. We need to be very specific. And this is not my field of expertise, but I know certain aneuploidies are known to be linked to early miscarriages with sort of unknown placental phenotypes. And I think we can now start looking at how different sort of aneuploidies are affecting like the early placental maturation, both in these extended cultures, but perhaps even more easily now with the human embryonic stem cells now even primed with certain sort of aneuploid changes, and then in vitro differentiate them into cytotrophoblasts and cytotrophoblasts and EVTs and sort of see is there a specific compartment that is affected by these different aneuploidies, and we can extend this into going into defects in the amnion and sort of seeing if there's certain sort of aneuploidies or changes that are linked to sort of failure there also. I think it's an exciting development.
Yeah, absolutely. I completely agree with you. Yeah.
So we have another question from the audience. It's from Bruce again. Bruce is being prolific today again. His general question is, in learning how to positively affect fertilization and implantation, how can we learn about potentially negative consequences, such as suppression of zygote loss due to genetic or epigenetic abnormalities that could lead to adverse fetal postnatal consequences? So I think what Bruce is trying to ask is, how can we learn from all the models we've been talking about today? How can we learn about negative consequences from those? So is there any kind of biomarker sets that you think we could develop? Do we need to go to extended culture models, for example, and study genetic and epigenetic normality or abnormality to understand fetal or postnatal consequences?
I think potentially we could tap into pre-implantation genetic diagnosis procedures where biopsies are taken from the trophectoderm, and with increasing sort of single cell technology, both at transcriptional and genetic level and epigenetic level, we could start biopsying or biobanking actually like half of the biopsy.
That's very small sample size.
And then sorry.
Yeah, so I'm just wondering.
Yeah, but if you have, usually you have somewhere 5 to 10 cells, and
up to 20,
that is now where people start splitting that. So you take like 3 cells for your actual analysis, and the rest you keep, and you can do another analysis, and then you can link that to different sort of types of pregnancy scores, or actually looking at after birth, I guess also. But start collecting that type of information. I don't know if it's being done already, but I think that would be extremely interesting when you start generating bigger numbers, because I think we need big numbers. It's not enough to have these small cohorts that we generally work with. But if you start tapping into all the PGDs that are done in a clinical site or multiple clinical sites, you might be able to start pulling out something that is significant and interesting.
The one thing maybe to add, Frederic, is, so of course, you have an epigenetic defect then the manifestation of that epigenetic defect can be much, much later than what we're talking about here. Much later. Can be in fetal development, can be in postnatal development, etc. So I think that's also worth keeping in mind, that epigenetic defects can manifest much, much later down the line.
I have another question from Bruce, but I'll go to Bruce's question next after this one. Can you develop embryo models of the BBSome cilia communication involved in the process of compaction and evolution and differentiation, or can they be developed?
Sorry, can you repeat that once more?
Yeah.
Can you develop?
Yeah, sure. Can you develop embryo models of the BBSome?
BBSome, what?
I'm not sure what BBSome is. I guess that is cilia communication, basically cilia communication involved in the process of compaction and evolution and differentiation. Maybe the questioner can fill us in on what that means, and then I can move on to the next question and come back to this one. Bruce asks, for early epigenetic methylation pattern regulation, how dependent is this on each cell cycle replication event? Are there specialized transcriptional regulatory machinery is coupled to each cell cycle dynamics, and how does tech couple to that? And that's a question, I think, for the panel. So how dependent is early epigenetic methylation on each cell cycle or replication event? In other words, do we see methylation patterns changing as the cells mature? I think that's probably for Wolf. And is there specialized transcriptional regulatory machinery that's coupled to cell cycle dynamics? And how does tech couple to that?
Yeah, so I guess that's a very general question in epigenetics, maybe, so I can answer it in quite a general way. So the key with epigenetic information is that it's transmitted across generations, either of individuals or of cells in this context. So basically, when a new DNA replication cycle happens, then the epigenetic information needs to be transmitted to the next generation. And there are many understood and then other not understood mechanisms of how this happens. I don't know whether the question is geared at that, but basically there are machineries like DNA methylases that copy the epigenetic information across the cell cycle. That's true for DNA methylation. That's potentially true for K27 trimethylation, potentially true for K9 methylation. Yeah, so I'm not completely sure I'm answering the question correctly, but it's absolutely essential that the epigenetic machinery integrates with the cell cycle.
I think we're down to our last few minutes, I think, at this point, aren't we? Or do we have a little bit more time?
We have a little bit more time, about 5 more minutes, Tia.
Yeah. So Wolf, for the side-by-side Soq-Fish analysis of transcription factor expression and binding motif availability, Gary wants to know what's the interpretation of the additional motif areas, because there were some locations that had motif available, but no transcription factor expression.
So that was very well spotted, Gary. So firstly, we have to be cautious about the technologies here. So could this be differential detection limits for the 2 different measurements? So that we have to be cautious that that could be the case and rule that out first, I think, before getting too excited about it. But if we allow ourselves to get excited for a moment, then there could be time delays. For example, the expression of a transcription factor precedes the chromatin opening. There could be cofactor availability. For example, there could be cofactors that are needed for chromatin opening, which are present in some tissues but not in others. And then there is another sort of smallish list of other things that could be happening there, all of which are super, super exciting, but we need to see the limits of the technology first.
And I have to thank the questioner. The BBSome, the defects in the BBSome induce the Bardet-Biedl syndrome. It's a set of genes involved in developing cilia on the cell surface, used to communicate and transport small proteins of information with other cells in its environment. And so the question was, can you develop embryo models of the BBSome with using the ciliate communication mechanism involved in the process of compaction and evolution or differentiation of the cells? And I'm not sure if anyone can answer that or has a question, has a comment on that.
I don't really know if anyone looked at cilia during early development of mouse or human. I do not know.
Is there any evidence that it's active in early compaction? We don't know.
I mean, I do not know.
I believe Kat Hatitonakis might have looked at that, and that primary cilia appear only later, not towards the end of, or early post-implantation probably, not during pre-implantation and not during compaction stages. I'm not sure.
I mean, cilia are important for left-right patterning, but that's
that's
much later.
I was going to say that, yeah.
I think we're, one question for Wolf, which might be able to be answered later, was a question on your figures. In figures 4.5 epiblasts and E7.5 mesoderm, what was the scale on the y-axis and what the units were?
Oh my God.
we don't have to answer that now. We can answer that later.
I'm
just saying that there is a question on that for you.
For methylation, it was probably percent CpGs methylated, genome-wide.
Mm-hmm.
But yeah, the person who's asking me the question can please, you can email me. I will try and answer the question as best as I can.
All right, thanks, panel. Do you have any final statements, or have we tapped your brains enough? No, I think, thank you very much. This has been a really great session, and it just shows how much more there is to do. All the work that you've done is enormous, and it just shows how much more we don't know. So thank you very much.
Thank you.
Thank you.
You can hand this over to Gary, I think.
Hi, everyone. I'm going to just share the closing slides. So thanks, everybody, for joining us for this human cell ALICE or HCA topic. And today's recordings, just a reminder, will be available within a week or two on YouTube and Bilibili. Thanks very much to Jean-Léon, Wolf, Esther, Frederick, and for the great talks, very interesting and extremely interesting panel discussion. And also for the all the people that are in the HCA Executive Office, Christine, Luke, Tracy, Sam, Jenny, Anya, John, Alan, and Yoshi for all the work making these seminars possible. And I should also thank Deanne for hosting this seminar series, or this seminar. And this is the last seminar of this longer series that we started in June 2020. We originally had a lung development seminar, and then we've had a total of 8 seminar topics that was created due to pivoting at the beginning of the pandemic. We originally had planned to have a development in pediatric cell ALICE meeting in Toronto in 2020, but unfortunately couldn't have that. So due to the pandemic lockdown, and so we created this online seminar series to make sure that we're not losing track of the science that we're all interested in. And we've had a number of speakers from all over the world in both Western and Eastern time zones, covering the topics, a number of topics relevant to development in pediatric development, early development and pediatric development. If you have any ideas for how these seminars can be made better, take the feedback survey that you'll see at the end of the seminar and will be emailed to you. And I wanted to mention some upcoming HCA events. There are 3 events currently on the calendar. There's another seminar series reviewing all of the biological networks. The next biological network for the HCA is heart and musculoskeletal. The next networks that will be highlighted are heart and musculoskeletal networks on May 13th. There's a large general meeting for the HCA in June, and relevant to this seminar series, we're finally going to have our HCA Developmental and Pediatric Cell Atlas meeting, August 25th to 27th, and I hope all the people that participated in this call can attend, participated in the seminar and attended, can attend that. Just a few more pieces of information about this meeting. The goal, this is the first meeting, that we're going to try and engage a diverse set of scientific communities to address major challenges in understanding development and pediatric development. You can see a recent preprint that is out discussing the human development roadmap and all of the challenges that we hope single-cell genomics technologies can help address over the coming years. We have a number of amazing confirmed speakers, Hans, Kat, Maz, and Barbara, and registration will open in June 2021. So please join the HCA mailing list and stay tuned on social media for announcements relevant to this registration. And that is it. So thank you very much again to everybody. And that is the end of today's session.